Okay, hello everyone. My name is Adel Salimi, and here is our a bit out of distribution presentation about improving gene expression quantification in real-time qPCR. Our method is called InterOpt, which is a combination of internal control and optimal. So the outline of this presentation, first I will explain how qPCR works and how we do normalization in qPCR experiments using reference genes, how we can improve this normalization. We propose 4 methods, and we provide some theoretical support for the proposed methods and their interesting features. Then the benchmarking and experimental results, and at the end, conclusion. So real-time qPCR is the golden standard for RNA and DNA quantification. They use it for the verification of the results after, for example, other NGS technologies like RNA-seq and so on. And it's because of high specificity, sensitivity, and low cost, and also the reproducibility of the results. But due to its technical errors, it's highly dependent on the normalization procedure. So the way the qPCR works, it uses repeated cycles of DNA amplification. So there's an initial RNA in the sample, and through cycles, it doubles, and when it reaches a certain threshold, which we call the cycle threshold, CT value, we can obtain the expression. And the cycle number which reaches there is the CT value. There are 2 sources of variation in this raw CT value which we get from the device. One is biological, which is what we are looking for, the biological variation of genes between samples. And there are also technical variations which we want to normalize them, and this cross-sample normalization is done using a reference gene, which is unaffected by the biological differences between samples. So the way you do it is so you have a target gene, CT value of a target gene. For normalizing its expression, you just subtract the CT value of the reference gene, so you have the delta CT. And then to compare this normalized value between samples, you just subtract the CT value, delta CT value of the target gene in, for example, 2 more normal samples. And this delta CT is the negative form of it is just a log-fold change, which you all are familiar with. And I should mention that there are different terms for reference gene. Like different gene, reference gene, housekeeping gene, endogenous control, internal control. I'm just using the reference gene word, and by gene, it's not just protein coding genes. It can be any sort of RNA molecule. So it's really important that choosing an unstable or differentially expressed reference gene, so we are normalizing our samples with, can lead to very contradictory results. I had an experience with a molecular genetics lab. They were trying to validate their experiments for a microRNA, and they used the U6 internal control, and they couldn't find any significant change in the gene. And when we investigated it, the problem was the U6 was differentially expressed itself, and actually, if you check other articles, still they are using this reference gene in different studies, although it's not a good reference gene to be used. And the main problem here is that there is no universal reference gene. Even the so-called housekeeping genes like GAPDH and HPRT1 have high variations in specific tissues or specific diseases. And for example, GAPDH and HPRT1 are differentially expressed in lung cancer biopsies, and or GAPDH is associated with drug resistance in prostate cancer. So the solution that they are using for this problem is using multiple reference genes instead of one, and also use study-specific reference genes. So based on the tissue or the disease, they choose different reference genes. So when we are using multiple reference genes, the conventional method is just take the average of, for example, this is there are 3 reference genes here in n samples, and they just take the average of the CT values, and you have an aggregated reference gene that you can simply use as a single artificial or virtual reference genes, like the reference gene, like the method I explained, the delta-delta CT method. So this is the conventional method. For decades it has been used, but no one has questioned it if this is the best way to do this, to just take an average of our reference genes. So could we do better? This is our main contribution. And the way we approach it, first we are answering 2 questions. The first one is, what type of functions can be used in this context? So the gene-independent technical variations, like the RNA concentration in different samples, is in a form of scaling operations for each sample. And the reference gene should preserve these very technical variations so that you can normalize it, normalize your target gene with it and remove the technical variation. So we present a family of functions. We call them scale invariant functions, which have this property that scaling the inputs is equal to just scaling the whole function. So it preserves the scaling of in different samples, which is caused by technical variations. And the arithmetic mean and geometric mean that are being used are also a family of this family of functions. So now we know what type of functions can be used for this aggregation. The next question is there a way to optimize them for more stable, and by stable I mean expression stability, into a single aggregated reference gene? How can we optimize it for our problem? And we utilize and when we say optimize, there must be some parameters. Here we utilize 2 other members of this family of functions, the weighted version of arithmetic mean and geometric mean. So now we have parametric functions. We can optimize them based on our objective, which is having a more stably expressed reference gene at the end. So we can normalize the data with it. Just like the example I explained in the previous slides, here just we are having a weighted mean. So each of the reference genes has a weight, W1, W2, and 3. And we are trying to optimize these weights based on our objective, which is the stability of the expression of this single aggregated reference gene. There are 2 most widely used stability measures are standard deviation and coefficient of variation. And we have also provided some theoretical support of why these 2 methods are good, the assumptions behind them, and also the reasons they are optimal for this problem. But because of for time management, I'm not going through that. So these are our objective functions to be optimized. And we have our two aggregation functions, our objective functions, standard deviation and coefficient of variation. And now we can propose 4 different methods by each of these 2 combinations. We call them arith-ST, arith-CV, arith-geom-ST, and geom-CV, which arith is arithmetic, geom is geometric. And we call these weighting methods. So when I say weighting method in the presentation, I'm referring to these methods. I should mention that there is only one previous work which is a bit similar. They have used a heuristic approach to give weights to each of the reference genes just by the ratio of their standard deviation. And we've explained its limits and problems in the paper. And in the figures, it is called GeoM-SDR. I'll show in the results section. So our first proposed method is the GeoM-SD method. And here is the objective function and standard deviation of weighted average. It is geometric, weighted geometric mean. And W is sum to 1. And actually, there is a closed form solution for this problem. And it simplifies to a regression problem, which has a closed form solution. And here's the solution. Next, the GeoM-CV method, we also try to find the closed form solution, but we couldn't. But we could calculate the gradient, the exact gradient for it, and we can use gradient descent to just to minimize it. So, and we used a special gradient descent method called Barzilai-Borwin here. So this was GeoM-CV, which is geometric weighted geometric mean optimized on coefficient of variation. Next method is ARID-CV. This one, interestingly, also had a closed form solution. Here is the objective function, standard deviation divided by mean. And the closed form solution is this. I'm not going into the details. And the last one was ARID-SD, which we couldn't find any mathematical solution for this optimization. So for this one, we use just exhaustive search, because the weights are just sum to 1. So you can, for example, for two reference genes, you can try different weights and find an optimum. So if I want to just give a summary about the method I just explained, here we are improving the normalization, qPCR normalization using weighted aggregation of multiple reference genes, and optimizing the aggregation weights in a weights. We are optimizing it in a way to minimize the biological variation in the resulting aggregated reference gene. Here I'm explaining another interesting feature we found in the GeoM-SD method, the solution of the GeoM-SD method. So I'm modeling the expression of two reference genes called X1 and X2, two random variables. This represents their CT values, and our objective was to minimize the standard deviation, and minimizing standard deviation and variance is equivalent, and so we just take the derivative of this objective function with respect to the W1 and W2 and set it to 0. It can be solved. Just I explained that it has closed form solution, and actually the weights that are calculated at the end are proportional to each reference gene's variance minus the covariance of the reference genes. So these are the solutions, and now I'm adding technical variation or noise to the expression of each of our reference genes. So instead of X1 and X2, I'll replace them with X1 plus F and X2 plus F. F is a random variable. It can have any distribution. And the interesting part is that if you plug it into the equation, this variable just cancels out. So the final solution doesn't change. This shows that because the solution is independent of the noise caused by the RNA abundance in different samples, which is our technical variation. And in conclusion, we can say that the aggregation weights that are calculated in GeoMSD method can be directly calculated based on the raw CT value, which includes both the biological variation and the technical variation. So now I'll explain the results. We designed a benchmark to evaluate these proposed methods and the previous works. The main point here is we use qPCR array datasets. These datasets have high number of genes or other RNA molecules, so you can normalize the data without a reference gene. So we have a golden standard for the normalized data, and we can compare this and calculate the measures on that. And our measure is the stability measure. The standard deviation and the coefficient of variation was I explained. There are two other advanced methods, G-norm and norm finder, which use some mathematical modeling for evaluating a reference gene. And by stability in the next figures, I mean is the average of z-score converted of these four measures. So we can just have one measurement and compare our methods. The data sets we used were two data sets, breast cancer and liver cancer. And one is tissue, one is plasma. And the main difference between these two data sets is the variability is different. It's much higher in the liver cancer data set. So we have different conditions. And so on the left side is the result for the breast cancer data set. The right side is for the liver. And this bar plot shows the mean stability of every two combinations of reference genes. So we just picked all of the combinations of genes on the data set, and we applied our method and other methods for normalization, and then we measured the stability, and this is the mean stability. And as you can see, the GeoMSD method performs much better than the conventional method, which is the red one. The green one is the GeoMSD method. All the proposed methods are better than the conventional, but the best one is GeoMSD. And we think that it might be because of the feature that I explained, the independence of the technical variation. And this plus is just a statistical test, Wilcoxon test, to compare each of these two weighting methods. And this shows that the GeoMSD is significantly better than other results. Another thing that we investigated is the sample size effect. So if we have smaller samples, a number of samples, then how would this affect our optimization? And it turns out that for lower number of samples, some of the proposed methods like the GeoMCV just get worse, and this red one is the usual mean, which isn't affected by the number of samples because it's not different in different conditions. And the green one is our proposed method, which you can see that still is better, it outperforms other methods. There's another point here that we have this dark green one is GeoMSD plus and the light one is GeoMSD. The difference between these two is we used some better estimators for covariance and variance to optimize our objective function. We improved the GeoMSD method for smaller sample sizes. So better estimators caused better performance for small sample sizes. Another thing that we investigated is if we can calculate the weights from an external data set, like an RNA-seq data set, and we just plug them to a qPCR data set. And I don't explain all the details from this. We have proposed how this can be applicable based on the number of samples. Which one is better if you use an external data set for the weights or just the actual experiment that you've done on qPCR. So I'll skip this part. And another thing that we checked was the effect of number of reference genes on the performance. And here the GeoMSD is the green one, and it improves until some point like 4 or 5 reference genes, but after that is a plateau. And this red one is the conventional method, usual average. But the point here is that in almost any case, you won't quantify more than 3 or 4 reference genes. So it shouldn't be a problem. And we think it might be due to some overfitting effect while the parameters go higher. So because more reference genes means more weight parameters. Now I'm giving you a real example of how actually this can be used in a real qPCR experiment. So here we have a breast cancer data set, qPCR data set, and adjacent normal. We want to quantify the relative expression of microRNA 25/5p between normal and tumor samples and see if there is any significant change. The reference genes are microRNA 16, microRNA 361, and RNU 48. This is not; these are not actual genes. We can say internal control. There are different terms for it, and these are selected based on literature and previously suggested reference genes. And what we can see in the results is that when you use the geometric mean to aggregate these reference genes, there is no significant change. But when you use the GeoMSD method for the aggregation, you can see this upregulation. And this microRNA is a well-known microRNA that is highly upregulated in different data sets. It has been validated previously. So this was a real example scenario for the usage of this method. To wrap up my presentation, we advise that using multiple reference gene is almost every time. It is very important in qPCR. It is required, and we try to improve this. In this case of multiple reference genes by optimizing a parametric aggregation function. And our main candidate method is the GeoMSD method, and it had really interesting features. We give some theoretical support for it that it's independent of the noise caused by the RNA abundance. It works on low and high variability conditions, works on small sample sizes, and also can be easily added to previously developed pipelines because you just replace the reference gene with a weighted aggregation method. We developed our package, which you can access in my GitHub, and it can be easily used for any qPCR experiment that you have multiple internal controls or reference genes. And also we provided some parallel implementations, both using CUDA and CPU. And you can also use it for finding the best combinations of reference genes among all combinations. So because it's parallelized, it can be run really fast. And obviously, I have not done this work alone. My friend Saeid Rahmani was the co-first author of the paper, and Dr. Ali Sharifi-Zarchi, my supervisor from Sharif University of Technology. And also, I should thank to Akamai Student Fellowship Program for covering the conference registration fees, and thank you all for listening.
We have time for a few questions. regarding like when you learn the weights for the reference genes, can you impose some sort of penalty or like sparsity to avoid the overfitting issue that you mentioned, and also like maybe learn a smaller subset of genes that can be used as a reference instead of like having to, you know, there's like the bias and they have to select which genes go to the weighting, and then you have to weight them and then use them in the testing procedure.
Yes, I think your suggestion is more applicable to account for other types of technical variations which are gene dependent, because in this case the gene independent variations is not affecting our solution, our optimization. But those cases may have some would be more affected with technical errors, and maybe regularization would help to avoid any overfitting or other problems. If I understand the question well.
Yeah, I mean, I just meant that, you know, by imposing
for the overfitting problem, yeah.
Sparsity, maybe you can select the subset of genes that are most informative for being a reference.
So in our case, we are actually trying all of the combinations for 2 or 3 and 4, all the combinations, because the genes are about a few thousand, and we are using CUDA and parallelization to calculate all of them. But for higher, maybe your suggestion for higher number of reference genes would be applicable.
So, question. So, how does this work relate to? You know, there's all this work which looks at batch correction. So, like methods like combat and RECOMB, which use housekeeping genes or reference genes to correct for batch effects in gene expression under the seq data. Do you think these kinds of ideas are relevant there, or is something different in qPCR?
Well, I think it might help. I'm not actually sure how the combat method aggregates the reference genes. I assume it is using also the mean of them, just taking average of them. I think this can also, this may also help to reduce the variability, technical variability, biological variability of the reference genes, and maybe it can, this optimization be applicable there too in RNA-seq. We actually tried using this method in RNA-seq to also find the best combinations, and mostly for generalization. So we pick the weights from the RNA-seq to another qPCR, not in your case, but in your case it will be just back to another RNA-seq data set. I guess it's worth to try this RNA sequencing and other expression technologies.